listen to it. And I'm going to share the screen. Yeah, you're able to see my screen?
Yes, ma'am.
I'll just make it big. Okay. So today we will go into, I think it is a very familiar chapter which mostly biotechnology students have gone through. But still, as it is in our syllabus, let us talk about this important aspect, that is monoclonal antibody production. We very commonly hear this particular monoclonal antibodies, especially when we talk about the diagnostic kits which are available these days. So we shall be talking about how these monoclonal antibodies are actually produced and what is the importance of these monoclonal antibodies in our what we call diagnostic, or as a matter of fact in any other what you call methods, especially in biotechnology or microbiology or diagnosis of diseases, or in any type of biochemical studies. Okay. Now, mostly when we see a perfect world where we consider that we are all hale and healthy, the microorganisms are very friendly with us, and no microorganism is going to come and attack you, because most of you are of, for example, if you take the present pandemic situation, most of you might be considering that COVID only occurs to the person next to you but not to yourself. Coronavirus is something which will not enter into my body but will only enter into a person body who is before me, isn't it? So that is what you consider it as a perfect world, that you are disease free. As long as you are not having that particular disease, you consider that you are not going to get that particular disease. But that is not so in the real world. In the real world, we have many number of microorganisms. I think if you have a type of a microscope where you are able to view these microorganisms, I'm sure there might be even a COVID virus just sitting right in front of you, or right beside you. Okay, so we have many number of microorganisms which are surrounding us, and there are chances that whenever, in case your immune system is not able to detect such pathogens, you will get that particular disease. So you have chickenpox virus, you have flu, you have what you call a common cold, stomach upset, so many number of viruses actually residing along with you. So that is how the real world exists. Okay. So in 1975, scientists of Britain Medical Research Council, which is considered to be one of the best medical council in the world, under the direction of Caesar Milston and George Kohler. So these are the two scientists who discovered a new technique for producing clones of antibodies using cells that synthesize antibodies of single specificity. For example, if you take your own antibodies, for example, take IgM antibody. IgM is an anti-immunoglobulin. I think you might be studying immunoglobulins, which Manoj sir is teaching you. IgM is a immunoglobulin. We consider this to be the first immunoglobulin to what we call to be synthesized, which comes first to attack that particular pathogen. So immunoglobulin by default will come and attack that particular pathogen. Immunoglobulin M is a polyclonal immunoglobulin. It has the capacity to eradicate or what we call counterattack many number of pathogens. Okay, but here we are talking about monoclonal antibody, very specific. That is, a monoclonal antibody of chickenpox will only attack the virus of chickenpox. It will not attack any other virus, even though it might be closely related to it. Such type of antibodies, we call them as monoclonal antibodies that synthesize antibodies of single specificity. Okay, they are known as monoclonal antibodies, and these were produced by this Milston and Kohler, who were awarded a Nobel Prize in Medicine in 1984. So they were awarded this particular Nobel Prize in Medicine for inventing or for producing this monoclonal antibodies. Now, what is the difference between monoclonal and polyclonal antibodies? When we talk about polyclonal antibodies, they are derived from different B cell lymphocyte cell lines. So they are derived from different B lymphocyte cell lines. Whereas monoclonal antibodies are derived from a single B cell clone. Okay, they are only produced from a single B cell clone. Whereas these are produced from different type of B lymphocytes. Next, batch-to-batch variation affecting antibody reactivity and titer takes place in case of polyclonal antibodies. So there is a difference when you are even producing them, there is batch-to-batch variations. Whereas in case of monoclonal antibodies, they offer reproducible, predictable, and potentially inexhaustible supply of antibodies with executive specificity. So these monoclonal antibodies have the same type of antibodies to react to a specific antigen. They are predictable, so you can say that these particular antibodies are what you call attack that specific antibodies. Specifically, they are very potential. They only attack such type of antigens. And inexhaustible supply. You can produce innumerable number of antibodies, monoclonal antibodies, and they are very, very specific. It is not that you produce a monoclonal antibodies of chicken pox, it will go and attack cow pox. No, it will only attack chicken pox antigens. Okay. Now, polyclonal antibodies are not powerful tools for diagnostic tests because they might give us some false positive or false negative results. Okay, so they might give us sometimes false positive and false negative results. Whereas in case of monoclonal antibodies, they enable the development of secure immunoassay systems. Very, very precise. If it is positive, they give it as positive. If it is negative, they give it as negative. Okay. Now, how is this particular monoclonal antibody production produced? Since its development, it has been very important in modern medical science with diagnosis, therapy, research, and even in basic science today. So, monoclonal antibodies are not only used for diagnostics, they are used in immunotherapy, they are used for targeting some type of specific cancer cells in the body. They are also used for even for blood grouping. The simple test which we do for blood grouping also uses monoclonal antibodies. It is still largely dependent upon animal testing, but still we are still depending upon animal testing. However, why? Because it requires immunization of mice in order for them to create antibodies of their own. So this particular monoclonal antibodies cannot be produced in vivo. You compulsorily have to inject the antigens into a mice so that the mice produces antibodies, and these antibodies are now used for the production of monoclonal antibodies. Monoclonal antibodies, or otherwise shortly called as MAB, is produced by cell lines or clones obtained from immunized animals with the substance to be studied. So, for example, you want monoclonal antibody of chicken pox. So you inject the antigen of chickenpox into the animal. The animal will produce antibodies. These antibodies are isolated, and out of which the specific antibody which is against that particular chickenpox is what you call selected, and these are made into clones so that you have a group of antibodies which are going to attack that particular antigen. Okay. Now, how is this particular monoclonal antibodies mostly done? The mouse is first injected with a specific antigen. So, for this, you require mouse, what we call the laboratory mouse. You have to have an animal house for such type of production. So, you cannot take a house mouse that is running around in the house and inject it with an antigen. You require a laboratory mouse. Okay, so you have to first inject it with a specific serum. Several days after inoculation, so what happens when you inject an antigen? The antibodies will be produced in that particular animal. Okay, after several days, maybe 7 to 8 days, the spleen is removed, because we consider spleen is the one which produces all such type of antibodies, and the B lymphocytes which normally migrate to the spleen. So first after several days, you remove the spleen, you castrate that, what you call, you sacrifice that particular mouse and take out the spleen that is present in that particular mouse, because it contains many number of B cells or B lymphocytes. Now you try to isolate such type of B lymphocytes, and what are you going to do? You're going to fuse it with cancer cells. So you are going to fuse these B lymphocytes with cancer cells, and these cancer cells are called as myelomas. Okay. Why are you fusing it with cancer cells? Because cancer cells have the ability to go on dividing without unproliferated cell division, we call it as. Okay. Myeloma cells are used because they can produce huge quantities of immunoglobulin and can live indefinitely in cell culture medium. So that is why you are going to use cancer cells. Because once you fuse it with a cancer cell, you can grow it in an animal cell culture. They will go on dividing as long as you are supplying it with the culture. So when you fuse these B lymphocytes with myelomas, we call it as hybridomas. The fusion results in a hybrid malignant cells called as hybridomas. So it's also called as hybridoma technology also. Okay, so first you are taking a mouse, injecting with a specific serum, antigen. We consider you are injecting with chicken pox. Okay, chicken pox antigen you have injected. So after a few days, what happens? Because you have injected the antigen, the organism, the mice will start producing antibodies against that particular chicken pox. All these antibodies which are produced by this particular mice, they will go and lodge themselves in the spleen. Okay, so they always migrate. The B lymphocytes will migrate, and they migrate to the spleen. So after that several days, you sacrifice the mouse and you take out the spleen, and now you are going to isolate that particular B lymphocytes and you are going to fuse them with cancer cells, myeloma cells. Okay. When you fuse this B lymphocytes with myelomas, we call it as hybridoma cells. Now, why are we using cancer cells? Because cancer cells have the ability to divide to any number of divisions. We call them as immortal cells also. Okay, so the fusion results in hybridoma malignant cells called as hybridomas. These cells can proliferate indefinitely and also produce large amount of homogeneous antibodies. So that is the reason why we are going to fuse them with cancer cells. Now, the cells can be grown in either of the two ways. By injecting into the peritoneal cavity of suitably prepared mouse, okay, that is in vivo or mouse ascites method, or by in vitro tissue cultures. The in vitro tissue cultures is the method used when cells are placed in culture outside the mouse body in a flask. So you can grow it in both the ways. Either you want to grow it as in vivo, that is you're going to inject it into the peritoneal cavity of the mouse, or you can grow it in tissue cultures, that is the animal cell culture tubes also. Okay, so this is a diagram which is showing you the production of monoclonal antibodies. So you have a mouse here. Okay, so you're injecting the antigen to this particular mouse. So it starts producing antibodies. Once it has produced this antibodies, these are the tumor cells, or what you call it as the myeloma cells. So you're going to fuse this cell into this particular tumor cell. This results in a hybridoma cell. Okay, so these hybridoma cells, these are the ones from where you are going to get your monoclonal antibodies, because they go on dividing for a very long time. Clones of the same hybridoma all manufacture antibodies of the single specificity called monoclonal antibodies, because they are derived from a single hybridoma cells. Because you are what you call injecting into the same hybridoma cell, the tumor cell. So when during that particular division, only monoclonal antibodies will be produced. As these antibodies are specific for a single antigenic determinant, it is more specific than polyclonal antibodies in recognizing specific antigens. Like I have told you, for example, you have produced, if you go to the previous one, for example, imagine this is what you call antibodies of chickenpox. Now you have injected, you have fused them with myeloma cells or the cancer cells. So this is a hybridoma cell, and these are the antibodies against that particular chickenpox. So this particular cell will be producing monoclonal antibodies of only antibodies which will go and attack chickenpox, but not to any other disease. So you can say that these are monoclonal antibodies against chickenpox. So they are very, very specific. So they have, that is what it says. All these antibodies are specific for a single antigenic determinant, and it is more specific than polyclonal antibodies in recognizing specific antigen. Example, a monoclonal antibody specific for cancer cell can be tagged with a radioisotope to detect a tumor in the patient. So that also can be done. So we said it can be used for diagnostic methods. So you can also use for the diagnosis of some cancer. So you can tag this monoclonal antibody to a radioisotope. So when it is injected into the body, the radioisotope will go and find out where that particular cancer cell is there, so that the antibody goes and binds to it, and you can get where that particular cancer cell is located. It can then be armed with a powerful toxin or chemical to seek the cancer cell and kill it. So imagine sending in a monoclonal antibody with a radioisotope and a toxin, so that this radioisotope will detect where that particular cancer cell is present. So along with the we are using radioisotope so that we can monitor where our cell has reached. So this monoclonal antibody will go and bind to that particular cell which is said to be cancerous. Once it binds, we have a powerful toxin, so that you can release that powerful toxin so that it will only kill that particular cell. This is very important, especially in treatment of cancers, because whatever we have treatments of cancers these days, it is either radiation or chemotherapy. And you all know that if it is radiation or chemotherapy, along with the cancer cell, even the normal cell also gets destroyed. Because a chemotherapy or a radiation therapy is not able to distinguish which is a cancer cell and which is not a cancer cell. That is the reason why we have many number of side effects when you are taking radiation therapy or in case of chemotherapy, isn't it? For example, if people are on chemotherapy, they have hair loss, they have what you call digestive problems, they have mouth ulcers, diarrhea, what you call loss of what you call white blood count falls down. So all these are some of the side effects which we are seeing, especially people undergoing this chemotherapy, isn't it? So in order to prevent such things, why not we use this monoclonal antibody? It will go and attach to only those cancer cells and then attack only the cancer cells, whereas the other cells are not even touched. So most of the side effects that might arise due to such type of what you call due to that type of therapy can be overcome. Okay, so it is then armed with a powerful toxin or chemical to seek the cancer cell and kill it. Now, what is the importance of this particular method? This method is used because antibodies must be what we call formed from immunization of substances being studied. So antibodies must be produced. Once the antibodies are produced, the animal suspect of the study can be eliminated and the tissue culture can be used. So first and foremost, you have to have an animal in order to produce the antibodies. Once antibodies are produced, you can go for in vitro cultures. When using live mice, researchers have found out that it is the better option because in vitro doesn't always produce adequate cell lines that are adaptive to tissue cultures. So they have also done experiments whether tissue in vitro is better or in vivo is better. Protein denaturation can occur from purification techniques and antibody activity is decreased with normal activity not represented. Also, cell lines could possibly become contaminated when using in vitro techniques. So these are some of the disadvantages in case you are going for in vitro techniques, that is outside the body techniques. So that is the reason mostly we go for directly injecting into the animal and then also injecting that particular antigen into the animal because this animal will now start producing antibodies. But when you are doing it outside, there are many problems which we can face. One is contamination problem. One is it or sometimes it does not produce adequate cell lines so that what you call you are not able to produce large number of such type of antibodies. And sometimes during purification techniques, because you know proteins are very heat labile, there are chances of protein denaturation also taking place. Okay, so what are the main steps of this hybridoma technology? The main steps of hybridoma technology or the production of monoclonal antibodies can be of first is immunization, then killing the animal and separating the lymphocytes. Third is selection of myeloma cells. Then fusion of separated myeloma cells with myeloma cells, sorry, separated lymphocytes and myeloma cells. Then selection of hybridoma cells and selection of monoclonal antibody producing hybridoma cells. So first is immunization. First you have to inject the antigen into the microorganism, into that particular animal so that it produces antibodies. Once the antibodies are produced, you're going to kill the animal and separate the lymphocytes from the spleen. Then you're going to select myeloma cells. So selection of myeloma cells, that is you can either induce tumor in some animals. So you can induce, you can what you call inject a carcinogen into a animal and induce tumors, or you can even directly take the cell lines which are available commercially from outside, which are called as the myeloma cells. So anything can be done. Okay, then what you're going to do, you're going to fuse these lymphocytes which you have isolated or you have got from that particular mice with this myeloma cells. Now, fusion does not always say you that gives you 100% of fusion. Not all myeloma cells will attach to all lymphocytes. Then you will get some fused cells, then you will get some unfused lymphocytes, and you have also have unfused myeloma cells. So you only want those cells which have been fused. That is lymphocytes which have been fused with the tumor cells. So then you have to select separation of lymphocytes from myeloma cells. Then you have to select that particular hybridoma cells, only those which have undergone fusion. And then you are going to grow this particular hybridoma cells in large numbers to produce monoclonal antibodies. Okay, so I'll be stopping my class till here. In the next class, we will go in detail, that is how this particular immunization is done, how we are going to kill that particular organism, how we are going to produce that what we call tumor in that particular organism, how we are going to select monoclonal, that is how do you know that monoclonal antibodies have been fused, any special medium we use in order to identify these monoclonal antibodies, and finally how we are going to isolate the monoclonal antibodies from that particular medium, and how we are going to use as a diagnostic tool for the synthesis or for the production of such type of what we call cells. Okay, students. So I'm going to stop share. Yes.